Hi everyone, today our group will present our mini project task 2, which is infographic about enzyme for our introduction to biochemistry subject.
My name is Mohammad Sharif Yusma with metric number E20191023500. What is enzyme? What is meant by catalyst? Enzymes are proteins that act as biological catalysts, also known as biocatalysts. Catalysts accelerate chemical reactions. The molecules upon which enzymes may act are called substrates, and the enzyme converts the substrate into different molecules known as products. Almost all metabolic processes in a cell need enzyme catalysis in order to occur at rates fast enough to sustain life. Metabolic pathways depend upon enzymes to catalyze individual steps. Classification of enzymes and its functions. First come oxidoreductases, catalyzed redox reactions, which means the transfer of electrons or hydrogen atoms. For example, alcohol dehydrogenase. Next, transferases, catalyze transfer of functional groups such as hydroxyl, carboxyl, amino, and etc. For example, hexokinase. Next, hydrolysis, catalyze hydrolysis reaction by adding water. There is one more types of hydrolysis, which is condensation reaction by the removal water. For example, trypsin. Next, lysis. Lysis involves the enzymes. This type of enzymes involve the cleavage of covalently bonded atoms, such as carbon-carbon, carbon-nitrogen, or carbon-sulfur, often to form a double bond. For example, pyruvate carboxylase. Next, isomerases. Formation of optical or geometric isomers, which basically means the rearrangement of atoms within a molecule. For example, malate isomerase. Last, ligases. Ligases involves the hydrolysis of high-energy phosphates, ATP, to form new bonds. Ligases is important in DNA replication. For example, pyruvate carboxylases.
Hi everyone, I'm Tiara and I'm going to share with you about unique properties of enzyme. So basically there are 4 unique properties of enzyme, which is higher reaction rates, milder reaction condition, greater reaction specificity, and capacity for regulation. So enzyme can help higher the catalytic efficiency, which 10 to power of 3 to 10 to power of 17 more faster than the corresponding uncatalyzed reaction. If there is no enzyme in a reaction, it still can occur but slow. Next, catalyzed reaction occur under mild condition, which is temperature under 100 degrees Celsius, ambient atmospheric pressure, and physiological pH, which is neutral pH. This is very useful in industrial application, which can help save a lot of money because we don't need additional energy to increase the temperature or to increase the pressure. Okay, so next, enzyme is specific in action and react with one or only few types of substance. Enzyme has their specific site or active site that can only combine with their specific substance. For example, enzyme arginase specific only to hydrolyze arginine. Last but not least, they can regulate biochemical reaction. This is necessary to prevent the excessive use of resources. So actually, enzyme can sense when to continue synthesis certain molecule or when to stop the production. It is depend on these two regulatory process, which is allosteric control and negative feedback inhibitor. So for this allosteric control, we can see this is an enzyme. There are two sides, which is one for the substrate and one for the inhibitor. So once the inhibitor bind to the enzyme, it is result in change to the active site mean for the substrate. So the substrate cannot attach to the active site and no product will be produced anymore. So for the negative feedback inhibitor, the end product will be the inhibitor and will combine to the enzyme 1. So initial conversion to end product will no longer occur.
Hello everyone, I am Kenisha and I am going to explain about how enzyme reactions work. Most enzymes are complex and has a three-dimensional shape which is very precise. Enzyme molecules are folded to form a cleft or pocket called an active site. The active site of an enzyme has a distinctive shape and charges that complement those of its substrate. Hence, the shape of the substrate must fit the enzyme precisely if a reaction is to take place. This explains why enzymes are very specific. The way an enzyme binds to its substrate can be explained by the lock and key hypothesis. The substrate molecule is represented by the key, while the enzyme molecule is represented by the lock. A specific substrate molecule arrives at the active site of the enzyme molecule. The substrate molecule that binds to the active site of the enzyme is known as an enzyme-substrate complex. The enzyme catalyzes the conversion of the substrate to products which are then departed from the active site of the enzyme. The enzyme is now free to take another substrate molecule into its active site to catalyze another reaction, while the product molecules leave the active site of the enzyme. Next is an enzyme regulation. Enzymes can be regulated by other molecules that either increase or reduce their activity. Molecules that increase the activity of an enzyme are called activators, while molecules that decrease the activity of an enzyme are called inhibitors. There are 5 major enzyme regulations. First is the feedback inhibition. The end product of a metabolic pathway acts on the key enzyme regulating entry to the pathway, keeping more of the end product from being produced. This decreases the activity of an enzyme, which is why it is an inhibitor. Second, cofactors and coenzymes. Many enzymes only work by binding to other non-protein helper molecules called cofactor. What is a coenzyme? The subset of cofactors that are organic, which are carbon-based molecules, are known as coenzyme. Thirdly, competitive and non-competitive. Competitive. An inhibitor binds to an enzyme and blocks binding of the substrate by attaching to the active site, while non-competitive is where the inhibitor doesn't block the substrate from binding to the active site but it attaches another site and blocks the enzyme from doing its job. Next is the allosteric regulation. The regulatory molecule, an activator or inhibitor, binds to an enzyme someplace other than the active site, which is known as allosteric site. Last but not least is the enzyme compartmentalization. Enzymes are often compartmentalized, which means they are stored in a specific part of the cell where they do their job. For instance, in a particular organelle.
Hi, my name is Noor Hafiza. So now I'm going to present you about the importance of enzyme. First, enzymes play an important role in metabolism, diagnosis, and therapeutic. All biochemical reactions enzyme catalyzed in the living organism. Level of enzyme in blood are of the diagnostic. It is good indicator in the disease such as myocardial infarction. Enzyme also can be used therapeutically, such as digestive enzyme. So next, we go to the regulation of enzyme in living organism. First, allosteric regulation. Second, activation of lectin enzyme. Third, compartmentation of metabolic pathway. Fourth, control of enzyme synthesis and enzyme degradations. And last, isoenzyme. So in conclusion, we can see that enzyme give a lot of advantage in living organism.